for the first 20 years of my life was education, basically. And then the next 30 years of my life was that I was a university professor, mostly at Columbia, but then at Berkeley, and then at Boston University, in many different fields in chemistry, biology, genetics, engineering. But then, roughly when I was 50, I switched to the private sector. And I co-founded a company called Sequenom, which became pretty successful, became a public company, and was the leader, at least at that point, in doing non-invasive prenatal diagnosis, and was eventually sold to LabCorp. And then about 15 years ago, with an ex-employee of Sequenom, I co-founded, and a third person, I co-founded a pharmaceutical company called Retrotope, which is very novel, and it's taken a long time, but I think at the moment we're running 3 phase 2 and 2 phase 3 trials, spending, I think, only about $60 million, which has to be a record. Currently, I'm involved in at least 15 different companies as an advisor, as a consultant, as a member of the board of directors, and so on. And a few of these companies are using artificial intelligence. Some of these companies are also connected to each other, so it isn't that many completely separate entities. And I must say that when I wrote a textbook on genomics in the year 2000, I had a section on artificial intelligence. All the way back then, there was one particular application which was very neat, which had been developed by some folks at Oak Ridge National Lab. And I devoted a significant amount of space to that, although it was not mainstream in genomics. I felt it would become mainstream, and I was right. But I think the important thing, speed is everything in drug development, for the reasons of intellectual property. That every year you get there quicker is one more year you have patent protection, and that's financially very important. And it's also important because the whole process is so expensive. But keep in mind that, I don't know, at least 90% of the cost of getting a drug approved for human use is in the late stage clinical trials. Okay? And what's embarrassing with so many of the hot looking drugs at the present time is they do okay in phase 2, but they fail in phase 3. And that's a terrible thing, because phase 3 is incredibly expensive. In some indications, the trials are $1 billion. And if you fail in phase 3, it's a disaster for the company, as we both know. Okay, so what my challenge to in silico is to do one of two things: either to figure out how to have less failures in phase 3 using artificial intelligence, or figure out, if you're going to fail earlier. In silico has managed to discover a novel target, and the target was a novel gene. Its function is not novel, but that's okay. They were able to develop some drug candidates for that target, and very importantly, improve the ADME potential of those candidates. That's a big accomplishment. What you've done with that drug by improving the ADME at a very early stage, that definitely decreases the probability of failure. Because as I mentioned, ADME is often where drugs fail in. Thinks artificial intelligence has a great future. It does. And for me, what's important is to do it without losing insight into the problem. It's very easy just to do it brute force to get answers, but there's no learning curve. And so I think I applaud what in silico has done with AI thus far, and I think it's only the beginning, though. It's not the end. Humans are not inbred laboratory strains. We're very diverse. When you have rare adverse events, which kill a drug, that's often, I suspect, rare genetic variations, which you had very little chance of picking up in phase 2, because the trial wasn't big enough. The other reason why drugs fail in phase 3 is you have all kinds of weird problems with ADME, again, individual variations, and you just didn't sample these sufficiently in your phase 2 trial. So what I'm coming down to, as our compute power every year becomes more and more powerful, I think it's going to be possible to take all the candidates for your phase 2 trial and model them, all right? And see which ones are likely to have problems, if any have problems, and either include them in the trial so that if there really are problems, you find them, okay? Or don't put them in the trial, but then you'll have a restriction on the label of the drug eventually. The drug will be excluded. Now, if it's excluding a drug for something rare that you can test for or model for, it's probably okay. In silico has made great progress on one of basically the major R&D challenge in pharma. Okay, but that's a small piece of the puzzle. I'd like to see you, because you have a very talented group of people covering lots of things, take it to the next stage and start to really make progress on the clinical trials.